I became somewhat of a submersible expert while I was in the military, right? So I got an edict from one of the 4-star admirals that said, hey, Commander, get up to my office. So what do you do when they say, Commander, get up to my office? You say, yes, sir. So the 4-star said, get up here. I ran. He says, look, here's what I want you to do. Here's $22 million. Go build me dry combat submersible. And I'm like, but. And he's like, Commander, which part of go build me dry combat submersible don't you get? And I said, yes, sir, right? So I had been in pressurized rescue module, deep submergence pilot. I'd been doing this undersea thing for a minute or two. So I have a lot of street cred in that, right? So with that, I have this strong desire to explore. So exploring for life is not just like a tagline. It's literally how I live my life. I want to explore for life, not for me, but for the next world that we're giving to our kids, right? That's the thing. We're exploring for the life of our oceans. We're exploring for the life of the generations to come. And we're exploring for our own knowledge. So you remember I told you I was a submersible expert. I get a call when I retire from the Navy from James Cameron's office. Now, James Cameron, for you young people, is a filmmaker, and he did a really good thing. He made Avatar, right? The new thing that he made was Avatar 2. But he made Titanic, right? So, and Titanic made my daughters cry. So he was like, you gotta go see James Cameron. I'm like, okay, send me a ticket. I have no idea if this is really gonna happen or not. Send me a ticket. So they send me a ticket. And I get out there, and I'm talking with James, with all these people about the 35,000-foot dive. I'm walking his dog on the beach, hearing the whole story of how he said, I got a good idea. I'm going to go to 35,000 feet. I'm like, oh, yeah, I had one of those guys tell me one of those things, and I got to go do something. Okay, great. So in this whole project, he goes down to the bottom of the Mariana Trench, the third guy in the history of the world to get to the bottom of the Mariana Trench, right? He finds this sea lice. You guys know what a sea lice is here because you're in Florida, so you get bit through your wetsuit, or they kind of get into your wetsuit and they give you a little nit, right? They give you a little anaphylactic reaction. You might swell up a little bit. No big deal. At the bottom of the Mariana Trench, that's how big they are. Holy mackerel. He had no idea what he was doing, right? So he just went, okay, I'll just grab it. Grabs it, puts it in his bucket, brings it to the surface. When we're doing the post-mortem on this, it's December 24th of 2012. I'm writing up the report. Why? Because it's James Cameron and it's due on December 25th. Don't ask, I have no idea. So I'm writing this up and I'm reading the overlay. And the overlay from the DNA sample is, this is a partial cure for Alzheimer's. Wait, let me say that again. A partial cure for Alzheimer's existed at the bottom of the Mariana Trench. Who knows for how long? Maybe even as long as Alzheimer's has existed, right? Holy mackerel. I sit back, I put my feet up on my desk, December of 2012. I said, we have to live in the ocean. Everybody's like, you're certifiable. Crazy, not gonna work. So I start, I'm retired, so now I'm doing other things. I'm starting to look at all these motivational quotes from people as I'm retiring from the Navy, and we didn't know how absolutely influential this quote was from Kennedy. Our survival may truly depend upon it, right? So if you think about it, if the origin of species is one thing, believable right now, right? I push the I believe button on that, please. If the origin of species is one thing, and I have a disease, the cure must exist somewhere because it all came from one thing. So if they all spread out, we have a disease, we certainly could have a cure. So he said, hey, let's stay underwater for 100 days. Now, this 100-day expedition did not just start with me going, hey, I got a good idea, I'm gonna go underwater. It went in 2012, I want to do this, and then I gathered together a team and we, come on down. Come on. I won't bite, I promise. So I got together a team. We're trying to work out all the logistics. It's actually very difficult. You have to learn all the systems. You have to learn everything to get down there. We started this in 2019 with a 5-person group. And 5 of us stayed underwater for 5 days. When we did that, we learned a whole bunch about saturation diving. I was a saturation diver in the Navy as well. And we learned a whole lot of lessons learned. And we said, okay, this is a lot harder than we think. 5 people is way too many people to put in this little box, right? So a couple of these lessons, and then COVID hit, and then you know what happened after that. And then by the time COVID kind of started ramping down, I said, we got to get in there. And everybody's like, I can't do it. My kid, my dog, my cat, my chicken. I'm like, I'm doing it. So they're like, all right, go ahead. So with their love and support, that's me. For 100 days, I went underwater from March 1st, came up June 9th. June 8th is what day? Does anybody know? World Oceans Day. No coincidence that my last full day underwater was World Oceans Day, right? We're trying to bring awareness. Remember, I'm a biomedical engineer. I'm not a marine scientist. I'm a biomedical engineer. So I stayed in this habitat right here. This was my teeny little bedroom, right? About 40 square feet, give or take. I'm going to go with a whole 7.5, 8 feet here. That was the bed for the visiting scientist. This is a wet room, as it says, and this wet room is that high. So you get to walk in there like this and jump in the water. That's the water where you come in and out of this place. This is a full height shower and a full height toilet, thank goodness, right? So the reason why it's bent over like that, because if there's any engineers in the audience, you could understand that a cubic foot of air has the buoyancy of 64 pounds. So basically positive buoyancy of 64 pounds. You need a ton of weight to keep this thing down. What they're trying to do here is cut the amount of cubic feet that you're using. Hey, I'm 6 foot. The guy's like, yeah, too bad, bend over. Okay, no problem. So then we're over here. This is basically 16, 17 feet long, about 8 feet wide, but it's a cylinder, right? So that means that you can't use a lot of this space, right? You could walk down the middle, and the middle, because the deck was so high, the middle was just about 6 foot and a little bit, right? So I was hitting my head on the ceiling at the first part. So there's the picture from facing in the kitchen, facing out my window. If you followed me on social and Dr. Deep Sea, you'd find that that was the place that I sat most of the time, right? All right, so why did I go underwater? What did I want to do? I'm a biomedical researcher. That's what I wanted to do, was biomedical research. I want to find out what happens to a human when you leave them in an isolated, confined, extreme environment. So here I am, stuffed in this tube, basically by myself, not totally alone, but basically by myself. Had some visitors coming through. We wanted to do some STEM outreach and awareness. I'm big on science, technology, engineering, and mathematics. I understand that there's another component to it, but I'm really big on this stuff. I really am, because we have real serious problems. And we need people to solve them. Researchers and scientists came to visit me, and I even had one of my old professors, an astronaut named Mike Gernhardt, Master's in Astronautical Engineering. Mike was my professor at Naval Postgraduate School, and he came to visit me underwater. He's like, hey, how are you? He didn't even know what he was doing. He came down, he's like, hey, now I remember you. So kind of really cool, but the good thing is that we talked about preserving, protecting, and rejuvenating the marine environment. We had all these ichthyologists, marine scientists, benthic geologists, people who knew about the ocean talking about preservation, protection, and rejuvenation. When I say rejuvenation, I mean regrowing coral. So we have the capacity. So I actually learned a lot while we were down there. So the biomedical research. Basically, this is a long-term study of what happens. We did 19 psychological and psychosocial tests, because you're in a little tube. Is there any other little tube that we might be in for a long period of time, going from here to maybe Mars, that we need to figure this stuff out now? Like, we haven't done real, true research in a long time in the water, that's for sure. Sea Lab was 60 years ago. It's coming up to be 60 years. Next year, it'll actually be 60 years. Technically, it's 59, right? So we did a whole bunch of testing. We did blood, urine, saliva, I mean, a whole bunch of real live testing and stuff that they didn't even have back 60 years ago when Sea Lab was a big thing. So here's the stuff that we did: full methylation panel, circulating stem cell, flow cytometry, the whole bit. I mean, we went crazy, $6,000 worth of testing if you're keeping score. Sleep studies, electroencephalograms, okay, so we're basically hooking up leads to your head. If you've never hooked up leads to your own head and done an EEG on yourself, you are missing out on life, man, because this is like an hour and a half of my day every time when I was doing it. It was horrible. Electrocardiograms on yourself, pulmonary function tests, many other tests. So basically, I did between 6 and 8 hours of science a day. No kidding, science, like work, right? And then about 3 hours or so of outreach, and then 2 or 3 hours of like media interviews, which totally shocked me. I had no thought or desire to be anything in the movies, but you know, now all of a sudden everybody wants to have you on camera, and I'm just like, okay, this is crazy, but this is the silly stuff that we did. So we got a bunch of the high school students in, and we brought them down, and you know, here we are drawing blood on them and looking at it under a microscope. Nobody had done that. Nobody had done that, so to our knowledge, and I have a great team of really experienced underwater explorers, to our knowledge, nobody's actually looked at it under a present-day good microscope. Like, we've seen it magnified, but not under a microscope. So we've seen some really cool things happening there. We did a whole bunch of ear experiments, including how when you go underwater and you stay underwater for a long period of time, the supposition is that you're in a warm, dark, moist environment, right? So if you're in a warm, dark, moist environment, what's going to grow? Bacteria, right? So we had to culture the ear on a daily basis. What they do in the Navy is they turn your head over and you put 50-50 vinegar and alcohol in your ear. You let it sit for 5 minutes. It's called dome burrow solution. And what it does is it basically cleans out your ear and makes sure you don't get an ear infection. Stayed for 100 days, didn't do it once, had 0 ear infections. Nah. You know, like you go back to the Navy, hey, why are you doing it? There's all these hypothetical things that could possibly happen, but they don't actually come to fruition. So lots of work, day and night. I worked like a dog. Well, so here's some of the results that have come out of this. So 50% reduction in every single inflammatory marker in my body. Holy mackerel. What does that mean? You guys hear cytokine storm? COVID, right? You're having a cytokine storm. You're in systemic inflammatory response. Cut in half when you're in hyperbaric medicine. Hyper meaning more than, baric meaning pressure, right? So we knew that. This is something I did not know. Humans generally sleep 30 to 33% of their time sleeping in deep and REM sleep. That's great. That's restorative enough sleep. You get 7 to 8 hours of sleep a night, you're good. You get that 30 to 33, you're good. Turns out, while I was underwater, I was sleeping between 60 and 66% in deep and REM sleep. Massive. I was like on fire while I was down there. So this was a totally unintended consequence. Cortisol decrease. Don't know why. And when I say cortisol decrease, I mean my cortisol dropped from the mid-80s, highest in the morning, right? So you have to check it in the morning. Peak scores in the morning dropped to single digits. Well, what happens when you have a decrease in cortisol? You have a testosterone increase that spikes through the roof. Holy mackerel, that's incredible. You guys mind if I take my jacket off? Okay, you feel free to take your jackets off if you're hot. I'm like, wow, it's getting warm up here. Okay, so that kind of stuff was happening. There are hundreds of other tests. So I am 50, damn it. See, doesn't matter how hard you work, I am 55.59 years old, not 59. I'm 55.59 years old. That's my real, actual chronological age. My extrinsic age, this is a calculation brought on by a whole bunch of external testing and a whole bunch of factors, is 34. Now, in earnest, when I went into the tube, when I went in to do the study, the week before the study, my age was 44. So this is a tranquil, steady dedication of a lifetime. This is spelling gym with a G and not with a J, right? Like I get up in the morning and I go to church, which is the gym for me, right? And that's part of my life. I eat fitness-wise. I'm always doing cardiovascular work. So this is, like I said, a slow pull over a lifetime. But what happened is I went from 44 to 34, extrinsic age. That's crazy. I mean, we're talking about telomere lengths, we're talking about things that are going on inside your body that are hallmarks of aging, like cellular senescence, all decreased from being underwater, all decreased from being under pressure. That was really cool. I didn't actually intend that. So here's just a flash of some of the data. I mean, hundreds of, so I have like 1, 2, 3, 4, 5, 6, 7 interns, and these kids do nothing but data pull crunch. They are amazing, right? So we go through all this stuff. But the good news is you don't actually need to do anything crazy like stay underwater for 100 days. What you can do is get almost the same or similar type stuff from using hyperbaric medicine. All right. So that being said, I want to talk to you a little bit about my second goal, which was STEM and outreach. So we tried to do a lot, and I think we kind of hit the nail on the head here. So when you reach 5,570 9 students and teachers and talk to them about science, technology, engineering, and math in a 100-day period. You're really reaching people. I did not anticipate, it was an overwhelming response from the teachers who really wanted us to come into the classroom. So that was kind of a cool thing for us. A bunch of on-site visits, and these are teams of people, not just singles. 9 people, 9 overnight explorer missions. Basically, those are 2 students sleeping in the other side, me and the adult sleeping in my side, and you know. So, but what you do is you get to form a bond. They get to become an aquanaut, and then they get me 24/7, right? So they're like, okay, hold on, I have a question. What's with the lobster? Why do they molt? How do they molt? How does that work? I'm like, oh, hold on, my friend Fred, the lobster who lives right outside, let's go look at him. And I literally saw Fred molt. I watched him molt. So I'm sitting at the window, and Fred is undulating. Well, undulating, he's basically doing this. He's kind of going like this. And I'm like, what the heck? You know, lobster, right? So I'm like, I'm gonna put on my scuba gear. I'm gonna jump out that hole in the middle, and I'm gonna swim out, and I'm gonna go see what's going on with Fred. So I come over there, and now that Fred is sitting right there. But right off here, skulking away in the distance, is a little pink gelatinous thing. And I'm like, Fred? He's not in there anymore. Fred just skulked away. He shed his skin. Oh, who sees a lobster molt in the wild? You might have it if you keep one in your tank, you may see one molt, but you definitely don't see one in the wild. See this cat? Anybody know who that is? That's Sam Patel. So Sam was Elon Musk's chief operating officer at SpaceX. We hired him. So we're doing cool things right now. Wait. So like I said, I got a bunch of visitors in the window, cool people coming by. I got to high-five everybody that came by. And I took it on as basically this is your job, right? Doesn't matter how you feel. Kid swims by the window, walk up and high-five him. Doesn't matter what you're doing, stop what you're doing and go high-five him. Why? Because we're incentivizing the next generation. We're incentivizing, hopefully, people like you into talking about the marine environment and thinking about the marine environment, right? Because where does most of the oxygen in our planet come from? You know the answer, the ocean, right? 60 to 70% of the world's oxygen comes from our oceans, right? So when you're thinking about it like that, and it's perfluorocarbon, I think is the exact name, these are the things that produce the most oxygen on our planet, but we need that oxygen. So we should just take this and I hate drinking plastic bottles. I know it's a necessity, but I hate them. I love reusable bottles. I was really thirsty. So when we take this and throw this in the ocean, we're polluting that ocean. So we really have to start thinking and taking better care of it. Because like they say, the solution to pollution is not dilution. So we learned a lot about preserving and protecting and rejuvenating the marine environment. We also learned a lot about science. So this right here, one of the people that came down is a microbiologist. I am by no means a microbiologist, right? So this cat came down here. He took a sample of the dirt outside on the floor, in the water, underneath the habitat. So it's basically just a mat of dirt. He took a sample. We looked at it under a microscope. That is a brand new species, new to science, a single-celled psyllid. I was like, what? He's like, oh, absolutely. Absolutely. Now we're going to have to write it up and do a bunch of tests to figure out, but I'm 98.97% sure. And I'm like, wow, that's kind of cool. So this is human blood underneath the microscope, which I thought was cool. So you get that biconcave formation of the red blood cell, which transports oxygen in your body, right? Well, when you're under pressure, what happens is, apparently, they stack up like quarters. This is called rouleau formation, and it is a predictor of decompression sickness when you're on the surface. Every single person underwater, their blood looks like this. How cool is that? I didn't know that. Nobody knew that. And I mean, we're talking textbooks. I literally have 5 chapters in hyperbaric medical practice. I know a little bit about hyperbaric medicine. This was not known before I went underwater. So that's kind of cool. So you see this? This is what everybody sees when they go scuba diving. When you go out and you go scuba diving, you see a whole bunch of fish. Anybody scuba dives? Yes, 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 yes. It's a whole nother world, right? Cool. I know everybody says that, but it really, truly is. This is my view from the top of the habitat. So when you experience life underwater and you're just a present day visitor, you run up and you look, you go, okay, oh, this is cool, right? So it's like meeting you guys. Hey, how you doing, right? And I fly by. Hey, how you doing, right? I don't know them, right? You got to live in the environment for a minute to see things, and you got to look closely. So here I am, a bunch of days into it, and I've been diving for 40 some odd years, and I've never seen a seahorse in the water. But I stopped, I slowed down, I looked, I became one with the environment. Here I am, 40 or so days into it, and I see my seahorse friend. I'm like, holy mackerel, I've seen a seahorse in the water. That is cool. Hippocampus, I'm all about the seahorse. Seahorse is the workhorse of the ocean. But you don't truly know a place. Like here I am, that'd be like interacting with you guys. Hey, where are you from, man? You're a scuba diver. Where are you from, right?
Missouri.
You're from Missouri? How is the person from Missouri a scuba diver and you people that live in Florida aren't? You're killing me, Smalls. So, but you know what I'm saying? So like you get to know this person just a little bit better, just like you get to know the environment a little bit better. Now fast forward to being underwater for about 70 or so days. You remember that little pool that I had in my habitat where I had to bend over to walk into it, where I get in and out of the habitat and go scuba diving from? That's the pool. That's a shark laying on the floor. She accepted me. I was there. She was all about having me in the environment. This is the difference between living in the ocean and visiting the ocean, right? It's like, you get this, and she's just like hanging out. I told her to put the cigarette out. She wouldn't put it out, so I made her move on. No, I'm kidding. So here I am. I surfaced after 100 days. So why did I say 100 days? I told you, right? I told you it's because I couldn't afford to stay 200 days. Why 200 days? How long is it going to take us to get to Mars? 200 days, guys. 200 days in an isolated, confined, extreme environment. So we need to figure out that science that helps you to survive in the 200-day go. That's just to get there. It's another year and a half to get back. So we're looking at a 2+ year journey to get to Mars and back, right? So we needed to do more time. I couldn't afford to do that much time, so I picked 100 days. Remember, the record was 73 days, so I had no intention of breaking a record. If I did, I have a life. So I would have done 74 days and then bailed. But this was not about a record for me. It was about setting a benchmark. It's about getting to a 3-digit number and setting a benchmark so that people would start to want to explore the ocean and see what's going on. That was me when I came out. Notice that I have all female doctors, all female physicians on my crew. They were absolutely amazing. So they worked me up and did some good things for me. This is the Association for Marine Exploration. So now what I'm going to do is I'm going to shift gears and I'm going to talk about the main impetus of this program, which is the greatest library on earth and why we need to save it. You're going, what? I came to talk about, no, I want to talk about diving in the mission. It's all the same. Don't worry about it. So this is a nonprofit organization, and that's their what they do. That's their mission statement, if they will. But don't worry about any of that. I'm going to tell you what they do. We're going to go over this in great detail. We do coral research. We do fish research. We do benthic geology. And then we do a whole bunch of medical engineering, biomedical engineering for me, and then a whole bunch of marine engineering, pressure proofing things to get to deeper depths. So that's the kind of stuff that we do in this organization. But let me talk to you about the thing that I talked about in the beginning. Remember I said for every disease there's a cure somewhere, right? We believe origin of species was the coelacanth, right? So if the coelacanth was that origin of species, in 4.5 billion years of genomic wisdom, we've come up with the thought that we have between 10 and 30 million species on this planet that bifarked from one thing. And if the disease is here, the cure is here. At least that's our thought, right? So, but regardless, each of these species has a story, right? Let's call it 20 million or so species. 20 million species is 20 million stories. So as you run down this road further, I'm going to talk to you a little bit about this type of exploration that we do and why. I'm going to flash through a couple of these real quick and get you to here. Whoops. I'm going to get you to right here, okay? So most scuba divers can go down to about 50 meters or so, give or take. You might be able to get a little bit deeper if you're really experienced. But if I rent a submersible time is un po' troppo cara. You know, it's expensive, right? So submersibles, you want to go deep and you want to look at all the cool stuff. Well, that leaves this area right here almost completely unexplored. Between 50 and 150 meters, all over the world is almost completely unexplored. Why? Because it's expensive and hard to get there. And if I had a submersible, I'm going to the deeper parts, so I'm not worried about this shallow stuff. Largely speaking, it looks like a twilight zone. Why? Because it's dark and twilighty, right? But unfortunately, we couldn't call it that because there are other zones in the ocean named twilight zone. Sunny zone, dark zone, twilight zone, abyss. Yeah. So unfortunately, we couldn't call it that. So we said, oh, deep coral reefs. These are kind of sort of deep coral reefs, but really, we really can't call them deep coral reefs because technically they're not. They're really, truly, you know, a deeper portion of typically shallow coral reefs. Holy mackerel, that's a mouthful. So then we went to the federal government for help. Don't go to the federal government for help because they'll give you this name. This is the name the federal government came up with was Mesophotic Coral Ecosystems, which is just about the least cool name I've ever heard, but that's now what the area that we explore is called. And how do we explore it? Like I said, most people will rent submersibles and they'll get in that little tube, right? I mean, that right there is basically a sphere, and this thing can go to, you know, most of the ocean depth, right? So here's my friend Tony Montgomery. Can you see him? He's in that little fishbowl. So Tony's now in the fishbowl looking out. He's like, I want to see fish. Actual picture, real guy, now has a PhD in marine science. At the time he didn't. I think he had a bachelor's or something, but he was really trying to do research. But that's how you do research if you have to be stuck inside that little tube. So we tried exploring. So now we're doing this exploration thing, and what we find is this. This is coelacanth, right? Coelacanthus lathus, it's actually the common name. There's another name for it that's scientific that involves a lot of syllables that I can't pronounce. We do a lot of stuff like this. I know you probably can't tell from this picture. On its right side, that is the deck of the Britannic, the sister ship of the Titanic, which sank in Greece. So we went ahead and dove on that, looking for stable climax, looking to do cool things. That's me on the propeller with the flag of the Explorers Club. That's me over there. So that's my gratuitous shot. So what kind of exploration do we do? I told you we do three kinds. We do coral exploration. Well, why? Well, coral is kind of cool. It's ubiquitous. It's all over the place. This is the kind of stuff that lives in our oceans that can adapt and thrive. So if it can live in our oceans and it can adapt and thrive, even though we're throwing garbage in the ocean, we probably want to learn a little bit about it. So this is one of our researchers way down at the bottom, and you can see the grand scale of this thing. So we're talking about deep, deep corals, right? Well, what we research is octocorals. Why? Because octocorals can survive in almost anything. Wait, octocorals can survive in a carbon dioxide-rich environment? Yes, they can. Is there any other carbon dioxide-rich environment that we're kind of heading towards if we keep messing it up? Yeah, so we want to learn how they improvise, adapt, and overcome. And they're 64% of all the coral in the ocean. So why do we call them octocoral? Why do we call them octopus? Shocker, there's the spoiler up front, right? Eight sort of fins, if you will. But why do we research them? Because they are high diversity. They're everywhere throughout the ocean, right? So global diversity, they're everywhere. The other thing is they have these little creatures that live on them. So these flagship species that we find on these fish, look at these cute little things, right? You know I'm all about the hippocampus, right? So I'm all about the seahorse. When we find a seahorse, we're digging it. So two size, can you see the seahorse? Right? So there's the penny, right? So we're not talking giant here, right? We're talking a baby tiny one. So you see that one, right? Can you see the second one? Right? It's neat. You got to look really hard. We didn't find it until we brought it back to the surface. And then we were like, oh, hey, look. Oh, a new species. Yeah, cool. So this is one of my researcher friends, Sonia Rowley. She's a meticulous diver, probably one of the best divers in the world. You know, here we are in, where were we? Ponape. And you know, she's a meticulous diver. We're talking, this is 135 meters. So here we are, look at it. You can see it's a little twilighty, right? It looks twilighty, so that's kind of why we wanted to call it the Twilight Zone, but it didn't pan out. So everybody thinks that we only just go and we go diving and it's all cool and then we kick on the beach. No, that's not the way it goes. When you get back to the hotel room, your hotel room smells like formaldehyde. You're dressing down the samples, you're writing up the samples, you're correlating the pictures that you took, marking the colors, everything, right? Like, this is science, no kidding. So the answer to why is all the rum gone is when you are on Ponape and you run out of formaldehyde because you find that many new species, you have to run to the one store, it's not like a store, it's the store on Ponape, and you go, hey, do you have any alcohol? And they say, yes, we have rum. Okay, so we bought all the rum out of the store. So why is all the rum gone? It's because Sonia ran out of formaldehyde. So if you look at this picture, flashing before your eyes are new species to science that we found. Every single one of these gorgonians, or octocoral, are new to science, right? And we discovered them. Remember, there's between 10 and 30 million species on this planet. So we're not discovering more and more and more. So we're trying to do our fair share of the work, right? Now, to me, they all look like coral. But to researchers like Sonia, like that live and breathe this, they look, oh yeah, it's totally a new species of coral. Oh, that's totally, and I'm like, looks like the same coral to me, but whatever, right? So then we do fish research. This is the HMACS Britannic, and as you can see, it reached stable climax. It's really at a point where it's thriving and surviving as a wreck, so good things. So what I want to do is I want to walk through this entire thing. I'm going to flash them through. All right. So this is everything, and if you look at the left-hand column, here's depth in meters. Here's stations. So that's the number of stations that we set. We were collecting fish. So this is the number of stations that we set. So here's the species that we collected, the number of species total that we collected while we were down there. And then there are the number of new species. So why is it 0 all the way till you get down to 45 meters? Well, because all those fish have been found. We've seen them before, right? But when you look where nobody's ever looked, you will find what nobody has ever found before. So that's kind of cool, right? As you go down, you see we're getting more and more new species, more and more new species. And then the number of hours that we spent are displayed there at that depth. And you can see that when we got down here, this is a very difficult dive. 120-meter dive is a very difficult dive. There are very few people that actually can do it, and there are much fewer that can actually work. And you can only spend about a half an hour down there, 20, 25 minutes down there is a reasonable time. You're looking at a 6-hour decompression, maybe 7 hours of decompression, depending on the temperature of the water, right? But look at the new species per unit effort. The deeper you go, the more species you find. Wait a minute. We're finding 27 new species per hour when we go to 120 meters. Crazy, off the charts, right? This is good stuff. So here, every fish that flashes before you is a species new to science that we have discovered. Right? It's because we're trying to get out there and look, right? We're looking where nobody else has looked before, and you'll find what nobody else has found before. So kind of cool. Like I said, I wouldn't know. They all kind of sort of look the same to me. They fish, you know, they got the little tail thing in the back and the little eye up front. But short of that, you know, somebody knows that they're different. Does anybody know how we determine something different or not? If something is a new species? We go before what's called the commission. The commission is for ichthyologists, ornithologists, you know, every single science out there. You have a commission, and they are made up of 9 individuals that are elected by their peers for the number of papers that they publish, their overall knowledge. I mean, these are like real scientists that are experts in ichthyology. There are 9 of them in the world, in the entire world, not the United States of America. We're not claiming it's the Super Bowl, right? So 9 of them in the entire world. One of our guys is on the commission for ichthyology. So when he says it's a new fish, I go, yep, sounds like it's a new fish to me. So benthic geology, this is the other form of research that we do. If you look at this, does anybody know what this is? It's a cut in the karst, or a cut in the actual limestone, if you will. right? This is all around the Indo-Pacific Rim. It's called a wave bench. And a wave bench is, what happens is the karst is here. It's basically a growth of coral and the water has gone down. So this limestone, if you will, as the wave crashes into it, it creates what they call a bench or a wave bench. This is a wave bench. All around the Indo-Pacific Rim at 240 feet. 240 feet is a wave bench. And we've known this, right? So what does that mean? The water level receded to that level and sat there and crashed, right? So that's how low the water level went during the Ice Age. Makes sense to me, right? We go to 410 feet all over the Indo-Pacific Rim. Same wave bench. Wait, what? 2 ice ages, 2 drops, precipitous drop, or 1, and then another one immediately following thereafter. Interesting. So what we do is we core sample, we do a whole bunch of benthic geologists. This was John Rooney, Dr. John Rooney, a benthic geologist who passed away on one of these dives. Now I only bring that out not to scare you, but to remember that it's difficult. This type diving is difficult, and I don't want you just running out there and going, well, the doc said that I could, do crazy stuff. Just remember the takeaway that it's really hard. Oh, by the way, if you ever want to attract a tiger shark, I don't know if you would or not, but if you ever want to, bang metal against metal. As soon as you go tink, 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 they're like, hey, what's up, man? You got something for me? Like, they are seriously attracted to the metal banging. Every time I bang one of those hammers, yeah, they came. So it's not just that. This is the stuff that John Rooney has taught me. While doing all of this research stuff, you just look. And as you look, you see this right here. This is a tributary. A tributary is when the water level receded, the water pulled out. When it pulled out, it had to go away in some direction. That's the direction that the water went away in, and it cut its path through, which I think is really cool, right? So he showed me this all over the Indo-Pacific Rim. I thought that was neat. Biomedical engineering. I am a biomedical engineer, so obviously we do a whole bunch of that. Every paper that you see here is a new paper talking about how to get human physiology to that depth. I mean, when we were doing this back in the 90s, this is something that nobody was ever doing, right? Nobody was ever doing dives sub 120 meters for sure, but sub 100 meters, right? We also do pressure sort of engineering. I figured that was a little bit beyond the scope of you guys, but we engineer rebreather systems so that you can live, breathe, and work underwater. That's one of the things that we do. So back to the greatest library on earth and why we need to save it. What's this? Library of Congress. Who said that? Thank you very much. No, because I really want people to know what the Library of Congress looks like. What do you think is in the Library of Congress? First-hand works, right? I mean, this is the type of library where first-hand works are, right? Things that are written on papyrus. Maybe something that Washington scribed while he was in the White House at some point, right? Jefferson, as he's writing, maybe the first draft of the Declaration of Independence, right? Who knows, right? Nuclear launch codes, the President's Book of Secrets. There's a whole bunch of things that are in that library, right? But these are all first works, right? There are no like, oh, there's 12 books of this and you're going to go check that out. That's not what this library is all about. It's very similar to a library that existed in, Alexandria, thank you. My goodness, I dropped the nickel there. So the Library of Alexandria, right at the head of the Nile, not Alexandria, Virginia, but the head of the Nile, Alexandria. And during the time of its existence, this was the repository for all knowledge. You wrote it on a papyrus, you carved it on a piece of wood, it went to the library, right? This is where the knowledge was kept. Right? And there was a big fire. Somebody had lit it on fire. It was a kind of a war thing. I don't really know the whole details of it. But a very famous general was tasked to put out the fire. He said, go put out the fire. He said, right, got it. He goes, okay, I'm going to bring all my people, and I'm going to go, you guys, I want you guys to all get buckets from the Nile and put out the fire. You guys run in to the library and grab one of the great works off the shelf and take it out and protect it. I said, okay. Now, I'm not trying to say doom and gloom like the world is on fire. Our world is not on fire, but we're not doing well. Okay? So you remember, if you're between 10 and 30 million species, that's between 10 and 30 million stories, and we could possibly be unlocking the key to the cure for Alzheimer's, cancer, whatever disease is affecting a person that you love, right? So like I said, we need to find out all those stories. So what are we doing? We're being the firemen. We're being the firemen on this side, and we're running in and we're reaching out, we're getting the species that are new. We're collecting them, we're cataloging them. We're not trying to rape the ocean of them, we're trying to get one. Why? Because we might not have the technology to pull the DNA just yet, but we will at some point. And when we can sample that DNA, maybe that's the cure for the thing that you have to, look forward to as you get older, right? So here we are, we're looking at between 10 and 30 million species. That's all of planetary biodiversity. And we have found less than 5 million species. Like, it's not pitiful, but it's really pretty sad. And what's happening in our world? We're on the precipice of the 6th grade extinction. I mean, look in the science books, it's there. We are on the verge of the 6th grade extinction. I'm not saying that bad things are gonna happen, but let's just say that the cure for cancer is in the duck-billed platypus, and the duck-billed platypus goes extinct, and we have no more samples of that. What do? We had the cure for cancer, and we let it slip through our hands. So here's our current glide slope. We're not doing so well, right? Because we're not throwing money at it. When we throw money at problems, we fix problems. Geez, we went to the moon, for crying out loud. So we can solve problems. So if we don't do something different here, we're gonna miss the opportunity to see something. So like I said, what's the problem with that? We need money. So I hate to be like, oh, money, money, money. I'm not trying to, like I said, doom and gloom, and I don't want your money, trust me, but I'll take it if you have any. So what I want to do is I want to enlighten you as to where your tax dollars go. And this is not to incite you, but to inform you. This is where your money is going, and I'm being very frank. This is 2017 numbers adjusted from 2012, okay? So in 2012, the National Science Foundation Planetary Biodiversity Index, a one-year effort, $3 million. That's righteous money for you guys, right? College students, woo, no problem. It's not good. It's not enough, right? The overall for that was a 10-year for the entire National Science Foundation Planetary Biodiversity Index, 10 years, $30 million. Shift in order of magnitude, the entire National Science Foundation Division of Environmental Biology budget, the entire budget of the National Science Foundation Division is $144 million. Single space shuttle launch, $450 million. Do you guys know what the space shuttle is? Please tell me you do. It's a big flying airplane that we used to send to space. We stopped doing that, but whatever. So let's go another order of magnitude or so, and let's talk about, So let's talk about the entire National Science Foundation budget, $7 billion. Boy, we are getting to real money now. $7 billion is a real budget number, right? Absolutely. What do you think NASA's budget is? Annually, 18. Now, once again, these actual numbers are 2017, adjusted numbers from 2012. So this was in 2012, but they're adjusted to 17. That's kind of a lot of money, but you can see that it is double the National Science Foundation budget. We are speaking very loudly about what we want to research and where we want to go and what we want to look at. And to me, that's unfortunate. So here's some more information for you. All right, the International Space Station budget, $150 billion. The International Space Station is reported to be coming off orbit in the next 7 years or so. So by 2030, they believe that the ISS is going to come out of orbit. There's some civilian things that are going to replace it, or so they think at this point. Here's the entire space shuttle budget, the entirety of the budget, almost $200 billion. That's a ton of money to be throwing at space. I understand the need to go look, and I'm not trying to say that it's a lost cause. There's some good stuff that came out of that, but here's the Apollo, $207 billion. Now, noble goal, but we took 12 people to the moon. That's what we got out of that. That and Velcro, because we got Velcro from the space race, right? So there's good things that came out of it, right? Velcro is important, but $207 billion? $7 billion? 207, 7. You get it, it's a problem. So here's what we got for that, all of those billions of dollars. We're talking an effort of about $500 billion, right? We got 656 people have gone to space. Not all of that was through NASA, by the way, even though NASA funding is in there. 54 suborbit, 602, they reached LEO, low Earth orbit. That's where we basically spin around in the International Space Station or just hang out. 24 traveled beyond LEO. That's the 12 that walked on the moon and the 12 that went to the Hubble Space Telescope. Now, one of my professors was the one who went up there to the Hubble Space Telescope, long, lanky guy, right? Tall, long, lanky guy. The first group of astronauts that was 6 foot or over was in that mission because they needed people that were long and lanky. The thing here is he said, look, Joe, the first time, you know, I'd been to space before on the International Space Station in low Earth orbit, but do you know that parking orbit that they have in Star Trek where they look down and you can actually see the entirety of the planet at once? He said, that's the first time when I actually felt that parking orbit. And I was like, oh, kind of cool. So you had to be far away from the Earth. Here are the people that have been to the bottom of the Marianas Trench. 2 in 1960, 1 in 2012, that's my buddy. And then another 25 from 2019 till now. That's because a guy made a submersible that can go to the bottom of the Marianas Trench, Victor Vescovo. By the way, another Navy commander. He designed that, and he basically wanted to get to the bottom of the Marianas Trench to open up the discoveries that we could have down there. 28 people have been to the bottom of our ocean, but 656 people have been to space. Like I said, I'm not crying boo-hoo, but what we do or don't do in the next 10 years is going to affect the next 10,000 years, says Sylvia Earle. So I believe everything we need is here on this planet. We just have to find it. And if you look where you've always looked, you'll find what you've always found. So you got to turn over another stone. You got to look in a place that hasn't been looked at before. So with that, I will take questions. But if you want the Neptune 100 research results, they will be available on the website. They're not available just yet because they're not done. But I'll be briefing them at the World Extreme Medicine Conference in Edinburgh, Scotland. By the way, if you want to go to Scotland, I will be wearing a kilt when I brief, so that'll be kind of an interesting thing. And if you have a person that has traumatic brain injury, please refer them to me. This is kind of the thing that I do. Like I said, we're at the same pressure. When I was underwater all those days, we're at the same pressure at which we treat traumatic brain injury at my clinic on West Shore Boulevard. And then if you want to contribute to veterans healing, let me know because we try and treat as many veterans as we can for free. So let us know. Like I said, I'm not necessarily trying to get your money, but if you screen that, you can get to the website and learn a little bit more about Dr. Deep Sea and what we have going on. And with that, I will take questions. Hey, you had your hand up first, and I was choking on water. I'm sorry. Yes?
I have 2 questions, but I can do one at a time.
Okay.
I know it would be hard because you can't do a lot when you're in the tube, but did you feel more well-rested?
Absolutely. 100%. So anecdotally, right, this is how we do science. So we do it objectively, like I can look at objective quality evidence. My watch says, my ring says, correlated watch and ring data, by the way. Well, Rolex doesn't say anything, but the watch that I was wearing at the time said something. So 60 to 66% and correlated to every single person that came down there. After the first day or so of getting used to the machine noise, yes, everybody slept between 60 and 66%. Everybody felt better. Felt. What does that mean? I have no idea. But you were more well-rested, that is for sure.
And then, do you know if there's anyone else who is planning on doing a 100 or more days underwater and doing the same type of research to see if it would have the same type of results?
Right, so good researchers, right? Good researchers always go, okay, you have an N of 1. That means the number of people in my experiment was 1. Okay, that's not a lot of fidelity there, right? You know, like clinical trials, phase 2D clinical trials are 420,000 people, right? I have 1, right? So what she wants to do is see if it's repeatable by another scientist. What I'm at the point now of doing is observation. And here's what I'll tell you. If you are a young person and you are interested in scuba diving and staying underwater for 100 days, I will help you. I will. I will. It's not a record. It's not my record. I want this to be broken. We've said 100 days because we wanted to go over a threshold. We wanted to kick down the door and say, let's start exploring the universe, right? Let's start here where we live. And when we do all this exploration, maybe we'll find some cool stuff. But I will help you. If you want to do it, you can get in contact with me, dituri@usf.edu. I'm in global. You can absolutely find me if you're a student. If you can't find me, I'm Dr. Deep Sea. You can Google that, and thank goodness we have a great team of people that were excited about putting things online for me. So, yeah. Any other? Yes, sir.
Yeah. Twofold question. Did your bone density and muscle mass change?
Ah.
That's one.
Let me answer that one first, right? So bone density, why do we have bone density decreases? When you go to space, you have bone density decrease because you're no longer stomping, right? The action of stomping, the weight that we have creates osteoblast. Osteoblasts build bone, osteoclasts break down bone. You have an excess of osteoclasts, they run around and they eat your bone, right? And you lose muscle, why? Because it atrophies, because you can't use it in space. I can lift 5,000 pounds in space, it's no problem. There is no like, let's go to space and lift weights. So what I did while I was underwater is, I did not have a bone density loss, but I did have a small muscle loss. But what I tried to do to combat that was one single thing. I used basically rubber bands, resistance devices that they have on the International Space Station to work out. And when I did, I used these cuffs. So they're pressure cuffs. And what these pressure cuffs do is they block off this artery and they block off this artery simultaneously. And when you work out during that time, same thing around the legs, and I did squats, but I did reverse squats using the resistance band, not using gravity, right? So I didn't experience much muscle loss, which was my hypothesis that I can get you to the point where you won't lose as much muscle as you do on the International Space Station. Why? Because we're going to Mars. And if we go to Mars, we're going to be 200 days, and I don't want to be, who's going to carry me out on Mars when we land on Mars? Who's going to be there to carry me while I'm on Mars? Nobody. So we have to have muscle. We have to figure out the bone density problem. So I did not lose bone density. That doesn't mean that it won't happen because I truly wasn't in space. I was in an analogous situation for space, but not truly. So yeah, so I did not lose a bunch, but we did gain, and that's kind of one of the things that we're doing follow-on. We're doing zero-G testing in November with those muscle cuffs and trying to use resistance bands in zero-G for the first time. We want to show that we can build muscle while we're spacing. You know, I don't hate space. I know I kind of beat it up. I don't hate space. I'm kind of jealous of the money that they get, right? Like $500 billion. What can we do with that in the ocean?
And quick question. So I assume that you sleep right now as everyone else. Right now.
Yep.
Normal life.
Yep.
So did your blood work and everything, cortisol, did it go back to the normal?
Not all of the blood work went back to normal, but the sleep stuff went back to normal about a week after getting out. I was like, oh man, it stinks. So, but you know, when you're under pressure, you have increased metabolism. I suspect that the increased metabolism led me to sleeping better. I don't know. Incidentally, in the first 20 or so days, I dropped, so I'm 220 pounds, I dropped to 198 and I said, can't have this, right? I was eating the exact same diet because I'm a creature of habit. I eat the exact same thing every day. I'm pretty lame. 4 eggs in the morning with a bunch of cheese, you know, some salad and protein for lunch, and then some protein and green thing for dinner, you know, like asparagus or whatever, right? So that's pretty much how I eat every day. I did that underwater. What I had to do was increase the amount of protein that I was eating because my metabolism was ramped up. So if you want to lose weight, go in the water. But I did. I lost 20 pounds, man. It's crazy. Yes. Dituri at usf.edu. If you're a USF student, if you're not, just look up Dr. Dituri or Dr. Deep Sea. Look up Dr. Deep Sea. It's easy. You can get that. But hit me up at USF and we'll do whatever you need. We'll talk to you about whatever. Questions? Yes. Twins? You're in the same shirt.
We're here. How does the pressure compare if you're say you're scuba diving at the same depth as the
Exactly the same.
place you were in.
Exactly the same. So I was there, and the water was here, right? So here's the water. Here's my metal part of the deck of the habitat that I was in. That water, this whole place has to be pressurized to keep that water out. So it's exactly pressurized to that level in the water. When you go out in the water and scuba dive, it's exactly the same pressure. You have to clear, but once you get in the habitat, you no longer have to clear. Interestingly enough, there's a system, there's a camera that points in the moon pool. That's the pool that you get in and out of the habitat from. There's a camera that points at it. After a while, I could feel a pressure change. I could feel when somebody would come up into the habitat. I could feel it before they even got into the habitat. That slow wave that moves in front of the diver as they're coming in, I could feel somebody get in the habitat. And before they got in there, I'd be like, hey, what's up? Be like, how'd you know I was here? I was like, felt it. So interestingly enough, the pressure didn't really change, but it was the pressure that I was at. Good question, though.
How does the lighting affect it? Because it seems like it was kind of dim.
Right, so what you're talking about overall is circadian rhythm, I believe, right? So daylight, night cycle, right? This is that serotonin production, then it gets dark and you start producing melatonin, right? So serotonin drops off, melatonin comes up, and you sleep. Interestingly enough, I could see a little bit of light and dark. That was my only clue, right? I missed 100 sunrises and 100 sunsets, and I am a sun seeker. Like, I'm that cat that will go and sit on Bayshore and watch the sun come up. I literally did it this morning. Kind of a cruddy sunset, but I was still. I'm sitting on a rock, and I'm watching something that is so far away that if it went out right now, we wouldn't know for almost 8 minutes. Holy mackerel, right? I get to see that every day. So I'm a sun lover. When I didn't get the sun, I felt a little sad, seasonal affective disorder, right? I felt a little sad in the beginning, but, you know, getting in touch with the kids, getting in touch with all the people, seeing all the friends, it wound up working out. So, good question. Good question. Circadian rhythm is important. Yes?
Could you feel any side effects from the pressure, like headaches or anything?
Great call, right? So, what happened to me overall? I'm 6'1". I'm now 6 foot and a quarter inch. So I lost 3/4 of an inch. Yeah, I know, but think about it. Think about it. Engineers, right? I'm being what? Compressed. So I'm in compression. What happens to astronauts? Because why? They're in tension. They're in tension. They're being pulled apart by the fact that they're literally got zero gravity rolling around the Earth at a great rate of speed, right? These people are basically being pulled apart. Astronauts grow about 1.5 inches. Apparently, aquanauts that stay 100 days shrink about 3/4 of an inch. I'm still trying to get it back. I'm hanging upside down. I'm doing yoga. So, yeah, kind of interesting. But yeah, so there was some other side effects that happened. Why is your vision 20/20? Because you could see an object 20 foot away like it's 20 foot away. You saw my habitat. There wasn't 20 foot of anything in there. So by the end of well, you saw the pictures of the habitat. You did, right? All right, so real short distance, right? Real short focal points. So the whole time, I came out of the water and I was myopic. So I had nearsightedness, right? I'm like, damn, this sucks. But it got better after a time. It got better after about a week or two, right? So all the blood markers came back up. I just did another round of blood today, and my hemoglobin's back up to 16. Interestingly enough, before I started, my hemoglobin was at like 15.5, 15.9, something like that, which is where I usually sit. Well, when you go to altitude, you go train a mile high, you're at a less of an altitude, right? So when you're at less of an altitude, it's a thinner atmosphere up there, it's harder to breathe, your body compensates, compensatory mechanism, your body compensates by making more red blood cells, which transport oxygen, right? So that's great, you have more red blood cells. That's why all these athletes go up to Denver and work out and train. It's like blood doping. And then they come down to Florida and they're like, yeah, man. And they got all these extra red blood cells. I went underwater, my red blood cell count went from high 15s to high 14s. Not a lot of drop, but it did in fact drop. I knew it was going to happen. I hypothesized that it would happen, and it did. So reduction in vital capacity, a couple of other things came up that I believe human tolerance for that pressure and that depth is about 100 days, but I'm still willing to help you break the record if you game. Yes?
What kinds of modifications did you have to make to the equipment as a biomedical engineer to handle the pressure?
Yeah. Nope. So here's what happened. I just bought the equipment and brought it underwater and went, Jesus, I hope this works. I'm not lying. That's exactly what happened. And I was like, yeah, my computer, this computer up here is the one that I had underwater. Like, for instance, now that it's open, I really can't get this touchpad to work, and I have to do the touch screen. It's just a little wonky, but yeah, the equipment has to be under 1.7 times the pressure, right? And all those fixed things that have air in them, like if this bottle went underwater, it would be squeezed 1.7 times. Why doesn't it squeeze if it's full of water? Water's incompressible, right? So if water's we are made of water, mostly. So we are incompressible, effectively. And if we get enough air in our lungs, then we won't compress at all. Human tolerance is 2,265 feet, something like that. It's well over 2,000, a little bit over 2,000 feet. So we can go deeper than that, that's for sure. Any other questions?
You talked a little bit about the funding structures for these other projects. What about the funding for Project Neptune?
Oh, yeah. So mad funding sources came out of the International Bank of Joe. You can see there's a little light right now, but I did, you're a researcher, you fund your own research, right? Look, if you believe in something, just get behind it and do it. Support it with your love, support it with your passion, support it with your wallet, and that's what I wound up doing. And then I got on board with Marine Resources Developmental Foundation, and I came to them with a whole bunch of money, and I said, is this enough? And they said, no way, it's not nearly enough, but please. And they said, I don't care how good your hair is, it's not going to work. So then they said, what you have to do is you're going to have to allow us to send kids down to you and stay on the other side, and researchers to stay on the other side. And I said, wow, it's concomitant with my entire philosophy of this whole thing. That sounds like a perfect idea. And they said, okay, we'll let you go. So yeah, funding shortages all over the world, but yet we have $500 million in space research. Shh, don't tell anybody. Any other questions? Yes?
How is this correlating with your genetic research?
Yeah. No, good stuff, right? So traumatic brain injury research is important. Why my traumatic brain injury is the best thing that ever happened to me. September 7th of 2021, I was driving my 1947 Chevy on West Shore Boulevard. 9 o'clock in the morning, somebody ran a stoplight, T-boned me. Out, right? So I'm a traumatic brain injury researcher with a traumatic brain injury. Woke up in the back of the ambulance. I go to St. Joe's, the hospital. The kid, and I'm being very little, looks like a child, is hanging over me, and he's the neurosurgeon, internist. And I'm like, oh God, I really hope you're a good doctor, right? And, we're working through the 8mm hemorrhagic stroke prefrontal cortex. Wait, actually, technically it was on the left side. So prefrontal cortex left side, so executive function. So I'm like, okay, we got to get into hyperbaric medicine. We got to do this. They let me out of the hospital 7 days later, and I started hyperbaric medicine and a bunch of other stuff. Turns out that we were able to cure it. Well, and I don't say that very lightly, right? Like, we were able to fix my own traumatic brain injury, right? So we treat at 1.75 partial pressure of oxygen, and that's right about where I was underwater. The findings are exactly correlative, right? It's exactly correlative to hyperbaric medicine and what we know about it. Every single inflammatory marker in your body will be reduced in hyperbaric medicine. It's one of the mechanisms of action. We increase the number of stem cells. We can multiply your stem cells by 8, which is pretty significant. Stem cells are the cells that you use in your body to make things. Now, don't be fooled by these exogenous stem cells or this outside the body stem cells. They inject those into you, and you can maybe uptake about a third or a quarter of them. Not a whole lot of those things actually get used because your body goes, what the heck is that? I don't even know what that is. And then they flushes it out, right? Or worse yet, it calls it an invader and starts to attack it, right? Even if you use your own platelet-rich plasma and you push it back in, it still gets partially rejected. Not fully, but partially. So we make stem cells that your body takes all of them and uses all of them, right? It lengthens telomeres. That's part of the thing that I wanted to do, was steal the resolve of the world to understand the mechanism of action of hyperbaric medicine. So that you can go, wait a minute, it reduces inflammation. Hold on. My aunt has Crohn's. Crohn's is a disease of inflammation. It's an inflammatory disease in the gut. I wonder if hyperbaric medicine can be used for that. Now, I don't go out too often. I'm kind of one of those guys that, they'll send me home when the sun's setting. So I'm out at a restaurant, and the server, not the waitress, but the server is like, hey, I have a nice pan-seared. Oh my God, it's you. And I'm like, what? She's like, it's you. Oh my God, you saved my life. Do you remember me? And I'm like, check. 21-year-old girl. I'm like, I have no idea who you are. She's like, I'm the girl from USF that did the study. And I'm like, oh, the Crohn's study. 38 treatments in a hyperbaric chamber. She was tied to a bathroom on Model. She was miserable. And Crohn's is a really debilitating disease if you have it. I mean, it really is, right? Cured. She's like, my mother was just talking about you at our family get-together 2 weeks ago. She's like, can I take my picture with you? And I'm like, oh my God. Like, it works when it works, right? So it's an anti-inflammatory, for sure. And it's effective, and she hadn't come back. And that was literally years ago. Literally years ago when she started that study. So it changed her life. So it has the ability to change your life, and it's natural. Guess what? The chlorofluorocarbon and the plants and the trees out here make the oxygen. So it's really a very natural thing. It's nothing that, we go, oh yeah, we got to put this chemical in your body. It's really going to help you, but it might kill a couple of things. Nope, none of that, right? You need oxygen to support cellular respiration. You need oxygen. Almost every cell in the body uses oxygen. Do you guys know what cell in the body doesn't use oxygen? What transports oxygen? The hemoglobin. Hemoglobin don't use oxygen. That'd be like me delivering chocolate. I'd be like, I'm sorry, you're not going to get all the chocolate you want. It's just not going to happen. I have a problem. It involves chocolate and shoes, but that's beside the point. Did I answer your question? Next. Yes.
So you saw the effects with your mission and also in the humor, like with the sleep and the helping the probes. Why? How does it lengthen telomeres? And what is the mechanism?
Oh, the mechanism of action of, so it's called DNA polymerase. And reverse transcriptase. You and I would need to sit down in a classroom with a chalkboard and a whole bunch of things. But there's a process that basically your telomeres, the end of the thing, the chromosomes, the end of the chromosomes, they look like little shoelaces. There's a process by which your body uses those every time you replicate that cell that it's associated with, right? Every time you use it, you lose it, right? When you're in a hyperbaric environment, it changes the sirtuin to the replicate mode, or not reproduction, but the fix mode, the repair mode. It switches the sirtuin to the repair mode, and it repairs. And there's nothing wrong with the cell, so instead of repairing, what it does is build. That was really overly simple. You're probably recording this. If you record this, I'm telling you, David Shaw will kill me if I give that kind of explanation, but it's all right, he'll get over it. Any other questions? Yes?
Did you do any kind of maintenance for your lymphatic system while you're over there?
Did I do any maintenance for my lymphatic system?
Massage or keep your sinuses clear?
Nothing. No. Literally, I did nothing that I didn't do on the surface. And that's important. When you do science, you want to make sure that, Now, I got a sinus infection. I cracked a tooth. I cracked a tooth on day 12. On day 12, I cracked a tooth, and everybody got together, 10 doctors, 2 psychiatrists, 1 psychologist, or 2 psychologists, 1 psychiatrist, and we're all like, okay, what are we going to do? And they're like, well, you can come up. And I'm like, well, that's not an option. Let's vote for something else. So we're looking, and it's like, well, suck it up, buttercup. You're going to be in pain for a little bit, and we'll make an appointment. So it's funny. I got out. I go, come up. I go to Orlando first. I get driven to Orlando. I spend a couple of days acclimating, learning how to drive again, and then came back to Tampa. Went to the the dentist, and he's like, oh yeah, you're going to the, what is it, an oral maxillofacial surgeon. And he's like, we are doing an emergency root canal on you today. He says, I can't even believe you lived like this for how long? I'm like, I don't know, 100 days. He's like, yeah, that was probably stupid. So lucky we saved the tooth. Things are good. They did the root canal that day, and yeah. So like you come up with these things. You get a little sick. So my sinuses got congested. I got really dizzy. One of the things when your sinuses get congested is you have what they call different baric pressure, alternobaric vertigo, right? So this ear felt like it was at this step. This ear felt like it was at this step. So I was doing this, which is great. So me and the bucket were hanging out for a minute. Oh, it was bad. It was a bad day. That was day 92. People think like, oh, did bad things happen? But bad things happen all over the place, right? You can't prevent that. One of the people that came down probably had a little sinus infection. I got a little sinus infection. It was bad, but what do you do? What are you going to do when you go to Mars? Yeah, okay, there won't be those microbes in there, but people still get sick. They carry sicknesses with them. Things happen. They cut their foot on something. They get an infection. Whatever. Lots of things are going to happen. I keep saying, Elon's great. He keeps saying, we're going to Mars, we're going to Mars, we're going to Mars. I'm like, hey, I have a question. What are you going to drink? And everybody's like, you can drink your urine. I'm like, well, okay, you can drink your urine, but you can drink urine. It has to be purified. There's a process it has to go through. Now, you remember I told you about osteoclast and osteoblast? Well, it turns out when the osteoblasts that build bone are not being used, they're urinated out. Well, that's a protein that needs to be washed out of your urine because you cannot re-drink that. So it's like, okay, now we got to filter all this stuff out. So there's what we call line loss. So you start with this much water. You come out and you could drink now this much water. And the next time this much water. And the next time this much. So you're losing a little bit every time. So there has to be a replenishable water source. I have another question. What are we going to eat? Oh, we'll just eat tuna fish. Canned tuna. 200 days. Just to get there. Canned tuna. I have another question. What are we going to do with the freaking cans? Right? Like we go shoot it out into space. Wait, what? Now we're going to pollute space just like we did to the ocean? Don't do that, right? Like we have to have real solutions. And these are what I'm trying to get people to think of. Like there are real problems. There are real problems on this earth. Let's fix those. Let's focus on those problems. Let's focus on that kind of stuff. So what else? Softballs, man, these are easy. All right, I don't want to hold everybody here all night. So if you have questions, I'll stick around. I'll chat with you outside or whatever. I don't know if we're getting kicked out. Or there is, you can go over to the exhibit. If you have not seen the exhibit, go. Because you stand in the exhibit and it moves you. And I mean literally moves you. It's kind of cool. So
Thank you.
you're very welcome. Thank you guys.